our next speaker is Dr. Chris Heery. Dr. Heery is a medical oncologist and a researcher at the National Cancer Institute who focuses on studying immune therapies for cancer patients, and he is also a member of the Chordoma Foundation's Medical Advisory Board.
Thanks, Josh. So it's a pleasure to be here with you all. I wanted to, as I was sitting and listening to we listened to a lot of these talks in some more detail over the last 2 days. And I was thinking about how a lot of you have come from far away to be here. And I think what you should maybe try to understand and try to give you a little analogy of where we are in medical technology today versus, say, 20 years ago. Imagine if you had to take your trip on a wagon pulled by a horse. That's probably where we were compared to the types of technologies that are available today, which allow the kind of science that you just heard about, which, to be honest, if you're a little confused by those things, don't worry, I am too. These are things that you have to go to very, very specialized places with incredibly smart people to even have them happen. And so I think we're lucky to have a leader like Josh who's able to pull these people in and get them involved in such an interesting topic and make them feel like part of the process. So I'm going to talk about immunotherapy. And you heard Dr. Flanagan mention that cancer cells are smart. I'm going to quibble with that. I'm actually going to say that cancer cells are just going through ultra-rapid evolution, right? So they are super dumb, but there's a lot of them. So you can think of them sort of like insects, right? And there are so many of them that you can swat one and you can swat another, but that's not really getting to the root of the problem. And if you put out pesticides, insecticides, you're going to kill. And I know I've talked about lawn care with one of my patients who's here today quite a bit. But you can put out things to kill weeds, right? Or you can put out things to kill a pest. And they will kill some percentage of them, but if you use them too much, they're going to get resistant to those treatments that you're trying to use. And so that's the complexity that we face. But sometimes, with these advanced scientific techniques, we can identify things that the cancer really needs to be what it is in the first place. And those are the targets that we want to go after. So I'm going to try to give you really basic tumor immunology lesson. So I really want anybody who doesn't understand any point to raise your, just go like, you don't have to do it high. So if you don't be embarrassed. But just try to wave at me or something if you don't get something, because the first few slides are really all that you need to understand to get to why we're doing some of the things that we're doing and what some of the side effects are and why we worry about just combining things without knowing what we're going to get, okay? So really do it, okay? Everybody got it? All right. Okay, so this is kind of the underlying problem with cancer immunotherapy, is that some cancers are already recognized really well by the immune system, and some are not. Okay, so the picture on the left is one that is not. It's just growing. There's no immune cells. It's not inflamed. And you might say, well, if the immune system recognizes the cancer, why didn't it get rid of it in the first place? How did it get to this point? And that is the point that we are going to get to. But for other cancers, they're not even recognized at all. And while cancers are different than the normal body, sometimes they're not different enough to be recognized, or sometimes they have changed through evolutionary processes to look just different enough that the immune system can't quite recognize them and interact with them. Okay, so I'm going to talk to you about some of those things. And I'm looking, any hands yet? Okay, good. So this is a really nice figure from a review article that was written a few years ago by some other really smart people. And they were trying to write this in a way for non-immunology cancer researchers to understand that this is an iterative process. And what I mean by that is that the immune system is a very smart system, and Dr. Flanagan could quibble with that, but so it is one that is able to pick up and learn things as it goes, right? So when you meet a new baby, what do you have to do? You wash your hands every single time you pick that baby up, and the mom says, no, you got to wash your hands, and then put the antibacterial stuff on, and then also don't sneeze or cough or get any. And the reason is that baby's immune system has not learned all the things it needs to learn yet, right? So over time, our immune system learns a lot of things, and you've maybe heard this, but it's been found through epidemiologic research that children who go outside and play in the dirt have less infections later in life and have less allergies because they have been exposed to more things. Now, they're probably sicker when they're younger, right? They have a runny nose more often, and it's harder for the parents. So I have 3 little children, and I know that sometimes I'm like, let's just leave them inside so we don't have to deal with them coughing at night. But it's important for your immune system to learn, and so that's why we have to interact with our environment to have those things happen. Now, so this is the process in tumor biology, okay? And because tumors aren't just like a virus or a bacteria that you might encounter, there are other ways that we need to kind of trick the immune system into seeing them when the immune system doesn't already see them, all right? So for tumors that are established, how can we get to the point that the immune system can learn about them? So let's start down here at point 1. This is what it says there in small letters, is that there needs to be a release of antigens, all right? So antigens, everybody knows what that is? Thank you. I knew you'd be the one. So an antigen, so you just heard about how our cells are made up of genes, and then those genes get turned into proteins, all right? And that takes a few steps, but those genes get turned into proteins, and proteins are what really make our cells what they are, right? So I say this to a lot of my patients, and some of you are here, so sorry, you've heard this before. But when you talk about a transcription factor, which brachyury is, it's an on-off switch for genes, right? So that lets certain genes be expressed in a way that you can actually see them turn into protein. And those proteins are what change the entire sequence of the code of DNA into something that actually means something. So, for instance, the DNA in the cells of the skin of my finger it's the same DNA that's in my eyeball and in my brain. And you might say, well, I used to think this. How is that possible, right? It's because you turn on and off different switches to make different parts of the code expressed. And once that code is expressed in a certain way, those cells take on the characteristics of the proteins that they make, okay? Everyone got that? Because that is a complicated idea. And I think as our technology of the world has improved to the point that now we understand sort of computer coding and things like that, we can understand how this is possible and how you might be able to manipulate these things. Anyway, so to get back to what an antigen is a piece of that protein that the immune system can recognize, okay? And antigens are little tiny snippets of the protein that have been broken down by our cells and presented on their surface in context of something else about our cell that tells the rest of our body that it is normal. And I'm going to get to what that is in a minute, so don't get lost in that. Don't raise your hand about that one yet, because I'm going to get to that. So antigens are little pieces of proteins, and they are unique usually to the types of cell. I am not going to finish all my slides because I want to really emphasize some of these understanding points, okay? So don't worry, you'll be able to go to the bathroom soon. So those are little pieces of the protein, and once they are released or passed on to the dendritic cells. So dendritic cells are a part of the immune system, and they're basically the cells that go around looking for antigens. And when they see one, it's their job to determine if it is a good thing or a bad thing. All right, so how does it do that? It's a complicated process, one that you don't, frankly, need to understand right now, but there are ways to trick the dendritic cell into seeing something as a bad thing. And that's one of the ways that we use vaccine platforms to trigger the dendritic cell to see something in a way that looks like it is foreign, meaning not part of the normal body. And then when it teaches in the next step, the T cells, it's going to say, I found this thing, it's bad, go get it. Okay? So the dendritic cells are the boot camp for these highly trained killers, T cells. And the T cells then have to find their way to the tumor, so they have to kind of travel through the body, through the blood. They have to get into the tumor and actually get into the tumor and start. Then they have to recognize the cell that they're going after, bind to it, and then kill it, okay? And on that last step, that very last step is where, if you guys have read the news at all about cancer research, you've heard about CRISPR and you've heard about immune therapy, right? And when people talk about immune therapy, they're mostly talking about drugs that target this step, number 7, the last step. And so when you see that, all right, 40% of patients with newly diagnosed melanoma can have their tumor shrink. Well, that's fantastic, right? So what's going on with the other 60%? It's something else on this the rest of these numbers, right? Something in there didn't happen correctly. There's also some other possibilities, but we don't want to get too complicated right now. So it is imperative that we understand how to drive this process in tumors where this last step, inhibiting it, isn't enough, okay? So that's what we're trying to do with Chordoma, okay? No hands, nothing. All right, good. So now we're getting more complicated, so we're gonna zoom in a little bit. And you're seeing that there are a few cells that are really important here. So the dendritic cell, I just told you about, is in green on the right. That's the one that is the training cell. It's gonna teach the T cell. The T cell's in the middle, and if you're a cancer immunologist, people call the T cell the center of the universe, and that's why it's in the middle in this picture. It's not the only thing, but it is important. And then there's some other cells that help regulate T cells down here at the bottom. Again, probably too much for us to talk about today, but this interaction between the dendritic cell, the T cell, and the tumor cell is really important. So I'm going to zoom in where this box is so we can look at that a little bit better. Okay, so this is the dendritic cell over here in green. And what you can see is that where it's interacting with the T cell, it has to teach the T cell about this antigen. Everybody remember what an antigen is? Okay, little piece of a protein, broken down by it and presented via this thing called MHC. Okay, so MHC is critical to teach the T cell. The T cell has a receptor called, and I think this is the most genius name, the T cell receptor. So it's easy to remember. The T cell receptor can only bind to antigens that are presented on MHC. All right? I'm going to say that one more time because it's really important. T cell receptors can only bind to antigens presented on MHC. Does it make sense? Anybody? Okay. So why am I saying that? Because a cell that doesn't present MHC is going to be tough to kill by a T cell receptor, right? And an antigen that can't be expressed on your MHC type, which are also called HLA, and if you've ever heard like, oh, I'm HLA2, I'm HLA24, okay, those are blood types. And they used to be, they're called human leukocyte antigens. And so they're the things that if someone's going to give you a blood transfusion, they're going to check to make sure that your HLA type is the same as the transfusion you're getting so that you don't get antibodies against that HLA, okay? So, because it turns out bad. So that MHC complex, the thing that's presenting the antigen, is actually fairly predictable. So if you're HLA-2, for instance, you can present certain antigens that someone who's HLA-24 cannot, and vice versa. Some of them are common, some of them are not. And so it's important that we understand that not everything that's going to trigger an immune response in person 1 is going to do it in person 2, right? So that's an important concept to get. So this interaction happens. The dendritic cell is trying to teach the T cell. They bind together. And then there needs to be this second signal as well. You see this, the pink one with the yellow. So don't worry about that. It's super, it's a little beyond where we're going to get in 20 minutes. But I'm only telling you this because these trigger points, these second signals, are also areas of active research to drive further activation of T cells in their interaction with dendritic cells, okay? But we're not going to get into that today. We'll be lucky if we get through the things I'm hoping to. So this now is what happens to the T cell once it's learned what its target is. It starts upregulating its T cell receptor. Remember what that's called, the TCR, right? And then it starts pumping out this cytokine. Anybody know what a cytokine is? I know Bill does. But so a cytokine is a signal between cells. And it's, if you took Latin, it's just signal cells, right? And so it's just telling another cell what it's all about, right? Or it serves some function to help kill or something like that. And this particular cytokine, interferon gamma, is one of the most important cytokines in tumor immunology. It drives our normal cells to respond to it by putting up this signal called PD-L1. And if you've read these things in the news about tumor immunology, it's PD-1, PD-L1. That's all you're gonna read about that over and over and over again. It's important to understand that that reaction is normal, right? It's something that our normal body does to protect itself from autoimmunity. Does anybody not know what autoimmunity is? It's exactly what it sounds like. It's the immune system attacking your own body. So if you know anyone who has lupus, rheumatoid arthritis, those diseases are autoimmune diseases where the body is the immune system is attacking the body. And this is a protective mechanism to prevent that from happening. So in tumor cells, this is why I say that tumor cells aren't that smart. They're not deciding to do this. Their genetic code tells them to do this. And in some tumor cells, their genetic code doesn't tell them to do this, or the translation of their genetic code doesn't tell them to do this. And so when you have a tumor cell that doesn't do this, it dies. And the ones that do it live. And so what happens is when you find a tumor that's advanced, metastatic, or very large, they are going to have some mechanism of resistance against the immune response in them. Either they were never seen, or they upregulated something like this. And so if you look through the literature in patients who have responded to checkpoint inhibitors, we can guess that probably the percentage of patients who respond had all the other things go right, and this was the only thing left, right? And so when we block it, instead of inhibiting the signal of the T cell that's trying to kill the tumor cell, we block right here, and now the T cell can kill the tumor cell. All right, that was a lot of stuff. Anybody want to ask a question before I go on to the next slide? Because this is really the crux of it. I could stop right now, and I feel like I accomplished something. All right, so I'm going to go on if nobody has any questions. So we're coming back to this, this idea that we can actually do something about the rest of this. And one of the ways that we can do this is actually with standard therapies. So this is releasing antigens. And some standard therapies do this really well. So you may think chemotherapy suppresses my immune system, right? Well, some do, but some don't. And some actually help kill cells in a way that those cells release information into their environment that the dendritic cells are going to pick up. And with that information, those cells are releasing little signals that say, hey, I'm bad. I'm not a good guy. And that signal is what actually lets the dendritic cell go ahead and teach the T cell. So certain chemotherapies do that really well. And radiation does it better than almost any chemotherapy. So it's interesting that when we think about the things that cure cancers, and I'm talking about other cancers now, but to some degree chordoma also, when we look at the regimens that we use in what we call adjuvant therapy, has anyone not know the word adjuvant? So it's when you do a surgery and you take out all the tumor and then you give additional chemotherapy. The majority of those adjuvant therapies have a chemotherapy in them that induces this type of killing. And a lot of them also get radiation, right? I don't think that's a coincidence. I don't know that anyone's ever going to prove that this is an immunologic event. But it's an interesting thought process, right? Like, is it that the chemotherapy is really wiping out every last cell, or is the chemotherapy helping kill a bunch of cells, and then the immune system's wiping up the rest, right? All right, so I wanted to just point out that there's one other thing, is that this MHC thing, the MHC processing and delivery of information that I talked about, that isn't always intact in tumors. In fact, in a lot of tumors, it's not working appropriately at all. And in chordoma, there is some research that indicates it doesn't work very well in chordoma. And so how do we get around that problem? One way is with another type of immune cell called the natural killer cell that can actually recognize and kill human cells that do not express MHC. So we have ways around this, and it's just about kind of thinking about each problem and then trying to attack it as it comes up. So for chordoma, this might be part of the solution, natural killer cells. And I'll come to this in a minute. Now I'm going to go pretty quickly through what we've done. Some of you may have seen this before, but I'm going to go pretty quickly, and you can stop me, raise your hands again if you have any questions. But this is one of the vaccines that we've used. It's a yeast-based vaccine. So what that means is we put the gene that we want, brachyury in this case, into yeast. We grew it up until the protein of brachyury is expressed. And then we kill the yeast with heat, and then we inject it under the patient's skin. And that vaccine, you might say, well, why does that work? Why would you put it in yeast? It's because yeast is already foreign, right? So the immune system's going to see this foreign particle and say, well, this is something that we need to deal with. I'm going to learn about this. And so within that foreign thing, we have brachyury, right? So now the immune system is associating brachyury with something bad. So we did a dose escalation where we gave increasing doses. We gave the vaccine once every 2 weeks for 3 months and then spaced it out. The safety of this vaccine was excellent, really just injection site reactions where we gave it just kind of like the flu shot. Although, honestly, my flu shot's always worse than what patients told me how this vaccine went, like 100% of times. That means I have a good immune response, though, so that's good, right? So in this study, we had a little more than half of the patients who had strong immune responses. And one of the patients, or maybe a couple of the patients that were on this study are here. And if that person wants to point out that he or she recognizes him or herself. So thanks, that makes it easier. I can just call you Bill now. So this is Bill's scan. So Bill had something unusual happen, which was that his tumors shrunk. And we didn't expect this at all. And it made us wonder if there was something different about what had happened with Bill before he went on the study. And actually, he had had radiation about 3.5 months before. I was truly a novice in chordoma at that time, and I didn't know if radiation was responsible for this. And honestly, I still don't know. But I can tell you that looking through the literature, seeing a tumor shrink this much with radiation is a bit unusual. So we were optimistic that maybe the vaccine helped make the radiation work better. And then we had another patient who I know isn't here, so doesn't have to call out or do a woohoo. And he went on the study after about 3.5 months after having this lesion on the left. So on the left, I circled these because MRIs are hard to see on the screen, even if you're a radiologist, just so you know. It's not you, it's me. So the one on the left is shrinking, you see, and and that one was radiated. But the one that wasn't radiated on the right is getting larger, and getting larger really quickly. So we radiated that one, and then this one shrinks away. And we thought, all right, well, this seems like incredibly good luck if this is 2 patients in a row who've had their tumor shrink with radiation and in a relatively short time period. So we thought, we know the vaccine is well tolerated, we know it's immunogenic, we know it's maybe doing something in the clinic. We want to answer this question of whether radiation is doing this or radiation plus the vaccine is doing it. And so we opened this randomized phase 2 study, and patients are assigned to either get standard radiation or radiation plus the vaccine. And so far we've enrolled 9 patients on the study. It does require that patients have only one site of disease right now. But I wanted to let you all know that this has become an issue with enrollment. We've had a hard time. We've had a number of patients who I think are good candidates, who make sense to be on the study, who have one little thing in their liver or one little thing in their lungs. And it just doesn't seem right that those patients can't be on the study, so we're trying to amend it. We're trying to change the study so that patients with a minimal amount of metastatic disease could be eligible. But in order for us to do that, patients with a minimal amount of metastatic disease, we'd have to make sure it's not going to hurt them in the near term, because we're not going to be addressing it as well as we'd like. So just something for you all to think about as you interact with your patient community, that this is a big change to the study. It's kept at least 4 patients in the last 2 months from enrolling on the study. And it doesn't seem fair. So we're going to try to change it. I'm going to mention really quickly that we also did another vaccine study with another vaccine targeting brachyury. We have collaborators in industry who make vaccines for us. This one was actually invented in our laboratory. I'm not going to get into details about it, but just so you know, by the end of, or the middle of 2017, we expect to have 3 vaccine platforms all targeting brachyury in the clinic. They will all be in trials. They won't be available to people not on clinical trials, but so far, this vaccine platform was also very immunogenic and very safe. The last thing I wanted to mention is that we think that we can use vaccines to trigger getting around this T-cell problem that might exist in chordoma. And it's a complicated mechanism, but we think we can use vaccines to help trigger an immune response that allows NK cells to be part of the solution. The last thing I wanted to mention is that anybody who ever wants to consider, if you have advanced disease or if you have a friend who has advanced disease and you want to consider a trial, we have basically made it a priority that in any of our phase 1 studies, so phase 1s are safety studies, that we aren't going to have the inclusion criteria written in such a way that patients with chordoma aren't eligible. So that was a weird way of saying that patients with chordoma are eligible for our studies. I don't know why I said it that way. So anyway, the point is, it's worth a call. Feel free to call me or my team and see if one of our studies is open and you might be eligible for those studies. So I just wanted to thank some of the stuff I've talked about. I've learned a lot from the people in our lab who actually do a lot of the science, and especially the Chordoma Foundation and these companies. Sometimes we vilify pharmaceutical companies, but these companies have been willing to let us put patients onto studies with patients with chordoma onto these studies, and that's I think a pretty big deal. So, and thank you to all of the patients who are here and have participated on some of the studies that we've been doing.
Are these vaccines good only for Seneca, or are they also good for?
So the vaccines target brachyury, and brachyury is expressed essentially in 100% of chordomas. There are some rare cases that don't express brachyury, but so they should be good for all chordomas. Yeah. Mm-hmm.
A few slides ago, you mentioned the other cancers that also were being treated with the vaccine.
Right.
Do they also have brachyury?
Yeah, that's a good question that we could talk about like for an hour probably, but but yeah. Oh, sorry. The question was, do other cancers express brachyury? And I think that's one that we should probably leave for now. That is why we developed the vaccines, because we believe that other cancers do. Yeah.
So Dr. Heery is going to be on the Ask the Experts panel in just a little while. So that's a great question to save for that panel. And just in the interest of time, if we can save questions for the panel, that would be great, and we'll go ahead and get the next speaker up. But round of applause for Chris. Thank you.